In chapter 11, now that we've learned about some of the characteristics of bacteria and microbes and all the other microorganisms, we're going to talk about how to control for them. So first of all, some definitions that go along with these, and unfortunately, in common terms, they get used interchangeably. So we're going to define them in terms of micro. So first of all, sterilization means completely destroying all microbial life. And the main thing, as we'll see coming up, is removing those endospores that some organisms can create. So again, being completely devoid of all microbial life is being sterilized. Disinfection, that's going to destroy most microbial life and reduces contamination, but this is on inanimate surfaces. So if we're disinfecting, like a hospital bed or a catheter or something to that effect. So it's an inanimate, it's not being used on anybody, but again, it's not sterilizing, it doesn't remove everything, but it does destroy most things. And then antisepsis, this is also called degermination. It's the same as disinfection, but we're talking about on living surfaces. So again, trying to remove most microbial life from a surface before like a surgery or a certain procedure. If that's being done on a person, then it's antisepsis or also degerming. So like using an alcohol swab before an injection, something to that effect. And then finally, decontamination or sanitation. This is talking about mechanical removal. So like washing hands of most, again, not all, not sterilized, but most microbes from either animate or inanimate surfaces. But again, we're talking about physically removing them rather than like some type of chemical treatment. Just a nice little kind of summary. It does look pretty busy, but if you can kind of walk yourself through this, it's a great study tool to kind of help organize all the information that we're covering here. So we have, we'll talk about physical control as well as chemical control of microbes, and then those mechanical removal. So mechanical, we're going to be looking at filtration, and if we're removing that from air, it's decontamination. But a lot of times liquids will be sterilized, so everything would be removed from them. As far as chemical agents, they're either gases or liquids, and again can be used either for sterilization or disinfection. On animate objects, that would be antisepsis. Inanimate objects, like on surgical equipment, now we're talking about disinfection or sterilization. And again, just your definitions here in this nice little table. And then our physical agents, this would be heat and radiation. And we have dry and moist heat. So dry heat would be incineration or a dry oven, and those can both be used for sterilization. Whereas with moist heat, if we're talking about like boiling water or pasteurizing for like milk, that's a disinfection, so it's removing most things. But in order to completely sterilize, there has to be steam under pressure. So we'll talk about an autoclave for that. And then looking at radiation as a physical agent, there's ionizing and non-ionizing types of radiation. Ionizing can lead to sterilization, whereas non-ionizing would be more disinfection. So again, remembering, first of all, knowing what these terms mean is very important, as well as all the different types of methods that are used. So this is a great summary figure to go through and understand and know. Now, the targets of microbial control, the ability is to prevent, microorganisms that cause infection, or with food and like pasteurization, spoilage. So they're constantly present all throughout the environment and the body. So it often also contains a mixture of microbes. So there can be bacteria, again, endospores, there can be fungi, protists, viruses, and prions. So again, we're not just talking about one specific thing we're trying to remove, but in these disinfections or sterilizations, we're trying to remove multiple different types of microbes. Now, if we look at the relative resistance, again, the most resistant form as far as living things are those bacterial endospores. So the bacillus and the clostridium organisms that can form those, that is the most resistant. In addition, prions, those infectious proteins, are also extremely resistant. And then we start going down, so mycobacteria, so organisms like that cause tuberculosis. They have a waxy cell wall that makes them really hard to treat. Others like Staph and Pseudomonas, Protis, and then we get down to kind of the least resistant being enveloped viruses, those animal viruses, and a lot of gram-positive are not very resistant because they just have that thick cell wall that can be destroyed and more easily targeted. So just kind of looking at this a little bit more. So again, those endospores, that's considered the most resistant microbial. Again, typically we're not worried about prions as far as disinfection or sterilization. So again, destruction of those is the goal for sterilization. So any process that kills endospores inevitably kills anything less than that. Then the other methods of control, like disinfection and asepsis, whether on inanimate or animate objects, this is just acting to destroy the microbes that are not quite as hardy as the endospores. Now, again, our antisepsis, first of all, just the term sepsis means the growth of microbes in blood or tissue. So if you've heard of septic shock, that's due to the presence of microbes in the blood system that create an immune response that is very typically life-threatening for the patient. So then asepsis would be the opposite. So a practice that prevents the entry of those agents into tissues to prevent infection. And then a lot of what you'll hear in the healthcare profession is aseptic technique. So these are methods that are used to exclude microbes. And therefore, antisepsis is the application of chemical agents or antiseptics that are exposed to the surfaces or wounds to inhibit those active or vegetative pathogens. So again, just some of our terminology that we'll use. A couple other terms here, cytal and static. So if we talk about a germicide or a microcide, these are agents that kill the microorganisms, whereas static, so bacteriostatic agents, they prevent from multiplying, but it doesn't actually kill the pathogen or the organism. So it's preventing the growth rather than killing them. So bacteriostatic agents are fungistatic. Again, they're inhibiting growth, but not necessarily killing the species. So some of the concerns that we worry about with control, things to kind of answer. So does the item require sterilization or is disinfection okay? So what's it being used for? So do endospores have to be destroyed in this circumstance? Is the item going to be reused or is it discarded? And if it is being reused, can it withstand the sterilization or the chemicals that are being used to disinfect it? And then, is the method suitable for the application? And again, could it be harmful to the patient? Is it too much or too little? And then, will it penetrate and actually get everything, especially worrying about biofilms? Can it actually destroy all of those organisms in the biofilm? Can it get into the crevices and wherever it needs to go? And then, is it cost and labor efficient, as well as, of course, safe to use it and for those and for what it's being used on. All right, so then there are some terms that go along with controlling on devices. So a critical medical device, that is one that's going to come into contact with sterile tissue. So it has to be sterilized. So again, back to our questions we ask about what type of method we want to use. Well, if the device is a critical device, it has to be sterilized. Semi-critical devices come in contact with mucosal membranes, so it may not have to be sterilized because that mucous membrane would help prevent any microbes from getting in to those sterile tissues. And then non-critical items are just those that do not touch the patient or only come in contact with skin. So like a stethoscope would be an example of a non-critical device. So again, depending on what the device is used for, would help us determine which control method to use. And there are a few different targets that can be used, whether we're talking about physical or chemical agents. So the cell wall, again, this is unique to bacteria. It's not something that animal cells have. So those can be used to either block the production, break it down, or just damaging it to make it fragile so that the cell lyses much more easily. They can also target the cell membrane. So loss of that permeability, especially detergents do this and break down the cell membrane with those lipids, but it also allows entry of other chemicals. And then the cellular processes like DNA and RNA, without those, they can't reproduce or make needed proteins. And then finally, altering proteins, so by either inhibiting ribosomes to make the proteins or denaturing or altering the function of the proteins already in the cell. This is just showing a picture. So normally proteins have the specific shape. So in this example, an enzyme is here, a substrate can bind to it, and it would work normally. That's called the native state. It can also change shape slightly that allow it to still be functional, but to where like the substrate can't bind. So it could still be active, but it's just in kind of an inactive state. Or substances could be attached that would block the binding of the substrate. So that would inhibit the protein from working. A lot of chemical or physical modes or methods of control, though, denature proteins, where it actually completely loses its three-dimensional shape and therefore becomes inactive. Surfactants, these are kind of like soaps. These are polar molecules that have both hydrophilic, water-loving, and hydrophobic, water-fearing parts. But these are able to actually physically bind to this lipid bilayer. So remember, all cells have a cell membrane with a phospholipid bilayer where the tails stick inward away from liquid and the hydrophilic heads stick out, connecting with water or other surfaces. So what happens is by adding these different surfactants, they embed themselves into the cell membrane and essentially just disrupt it. So it kind of pokes holes. It kind of makes these leaky spots in it so that, again, other chemicals then could get in and allow them to destroy the cell. So starting with our physical methods of control, again, from kind of the summary table, these include heat, whether it's moist or dry, and moist heat is the most widely used method of microbial control. Radiation, filtration, changing osmotic pressure, and then temperature the other way, so cold. These are all examples of physical control. And starting with heat, so that moist heat, this would be like hot water, boiling water, steam. Essentially what it does is it coagulates or denatures proteins very quickly and permanently. So then they can't go through metabolism, they can't get energy, and ultimately would lead to death. Dry heat would be things like desiccation or lyophilization, so like dehydrating, something to that effect, where it's kind of removing the water. So the air with a low moisture, like a flame or something, dehydrates the cell. So without the water, it becomes unstable. Again, those proteins don't work properly. And again, it causes the cell to become non-functional. Now it is less efficient than moist heat. It takes longer. So if you're putting something in an oven, it would take longer to disinfect than if you were boiling it. Another type of moist heat is pasteurization. And this is mainly used in the food industry, especially with beverages. So it applies heat to liquids that kill, mainly focusing on things that would cause infection or spoil the liquid without harming the flavor or the value. So it inactivates a lot of viruses and destroys reproductive, like vegetative bacteria and fungi. Again, not sterilization though, so it doesn't get rid of those heat resistant microbes or endospores. Now, the one that can be used for sterilization is moist heat or steam under pressure. So in this case, which is like an autoclave, the increased pressure raises the temperature, and that allows the temperature to reach heights about 121 degrees Celsius that are effective at sterilizing and removing those endospores. So this is good for like heat resistant materials and some plastics that can be used. Now, switching over to radiation, again, there's ionizing and non-ionizing radiation. But in either case, essentially what happens is energy is released at such high velocities that it ultimately destroys, especially the DNA. So it causes mutations or it alters the effectiveness of that genetic material. So this includes things like X-rays or UV radiation or even gamma rays. So it's mainly targeting that genetic material. Now, again, this kind of bombards at that cellular level. So again, ionizing is catastrophic mutations. So that is going to ultimately be used for sterilization, so they can't come back from that. Non-ionizing radiation, though, just makes it form some abnormal bonds or mutations that doesn't always lead to that sterilization. But in either case, we're changing the structure of that DNA. Now, another type, and this is one of the physical methods, is mechanical methods as well, is filtration. So this is actually removing microbes from the air or liquids. So it's strained through that filter, and then how much it's removing or how effective it is depends on how large of an opening there is. So in order for it to be used for sterilization, then those holes, again, it's porous, it would allow the liquid through or the air through, but not any endospores or microbes to come across. So HEPA filters are used a lot in hospitals that decrease airborne contaminants, prevent them from coming in, filter them out, especially in doorways. But again, also used in a lot of food industry, so like milk and beer, water purification are filtered. And as the picture over here shows, again, our filters here, depending on the pore size, again, the liquid or air comes through, but the microbes would be trapped and then therefore physically removed from the liquid or the air. Then we have our cold methods. So cold, of course, now we're talking about refrigeration or freezing. Neither one of these kill the organisms. Again, they're static, so bacteria is static. They slow down the activity, so prevents or slows their reproduction. But again, only some are actually killed by those temperatures. Most, though, are just adversely affected. So again, they can't reproduce, but then if it heats up, now they're then able to multiply and reproduce. And then finally, with our physical, the osmotic pressure can be used, and this is creating a condition. So if we have a cell here, there are always some different solutes inside of the cell, and then there are solutes or chemicals outside of the cell. Well, the way that water moves is it moves from where there's not a lot of these solutes or these chemicals to where there is a lot. So what happens with osmotic pressure, by adding large amounts of salt or sugar, so it increases all these solutes. So it puts a bunch of solute outside of the cell, and then what that's going to do is it's going to cause water to leave the cell and come out into what's called this hypertonic, this high solute environment. But essentially, it's a way of dehydrating the cell. So it loses its water. It can lead to what's called plasmolysis, where the membrane and the cell wall separate. But again, it just makes it unable to multiply. So things like cured meats or jellies or jams where lots of sugars are added to them. This allows things to be stored without refrigeration because, again, they don't have access to water, so they can't multiply.